So let me start by motivating the problem that I'm interested in. As probably many of you here interested in visual recognition, I'm interested in understanding the computational basis of invariant object recognition. So here's an example of such tasks. I can show you a bunch of images shown here, and I can ask you to tell me whether those images contain a specific object category, say an animal or a giraffe. I know you wouldn't believe the kinds of examples of giraffes that you find on Google when you search for giraffe online. And so I can imagine what the Google vision system actually learns about appearance of objects. Anyway, so this is obviously a very challenging problem. I don't think I have to convince anyone here in this audience. We've tried to engineer computer vision systems for many decades, and we still are very far from having computer vision systems that can compete with the accuracy and robustness of the primate visual system. So I think I have a lot of my colleagues in neuroscience actually complain a lot about progress in computer vision. I'm more on the optimistic side, and I think it's fair to say that computer vision has done a lot of progress in the past 10 or 20 years. I think you've heard in this summer school several examples about pretty ambitious and encouraging results, training very large neural nets on millions of images and achieving quite good results on a number of data sets. I think there are a couple of areas in computer vision for which we've done significant progress. Face detection is one of them. I think since the mid-90s, where the first systems were developed, we have now reached a stage where, of course, it's not completely solved, but we have systems that are useful enough, good enough, so that they could be used for kind of everyday real-world tasks. In particular, if you've bought recently a consumer-grade camera, you probably noticed already that there are built-in face detection systems that allows those cameras to make kind of smart adjustments, adjust the contrast and the focus of the camera onto what people typically care about in those photo shoots, about the faces that are included in those scenes. Another problem for which we've made great progress is pedestrian detection. So how many of you have heard about this Israeli company, Mobileye? A few of you? Okay. So this is an Israeli company that was created by Amnon Sashra a couple of years ago. Amnon Sashra is also faculty at the Hebrew University. What the company has been doing is build a small chip, which is here, that can perform reliably line detection and pedestrian recognition in complex visual scenes. The system is not all that exciting. I think it's an extension of some early work done at MIT in the late 90s. This is essentially an extension of R wavelets with some fairly fancy machine learning on top. What it took Amnon to build a system that actually works is a small army of engineers, about 100 of them, painstakingly implementing several heuristics to make sure that they reduce the levels of false alarms, but they did reduce the level of false alarms. So the company claimed that the system makes one false alarm every 30 minutes in a video stream for a hit rate which is close to over 99%. Okay? So that's quite amazing, especially given the fact that they simply treat every frame as independent of one another. So one false alarm for every 30 minutes is quite an achievement in comparison to some of the systems we have been developing in academia. So the system is reliable enough that when combined with a radar sensor, it's actually running on fairly high-end cars. I think it's already available on the Volvo S60, and I think the company says that it's going to be widely available in the next couple of years on additional car models. The point is that the system, again, it's not just vision, it's combined with radar sensor, is reliable enough that the company was willing to essentially hook up the brakes to the system, so that whenever the system detects a pedestrian at a distance which is too short for the driver to brake, the car will brake for the driver. So in terms of a Turing test for our vision system, I think that's, if a car company is willing to put their ass on the line to being sued, it has to be that the system is quite robust. Now, one issue with the system, and that's actually very well acknowledged by Amnon, is that it required several hundreds of millions of training examples to achieve this level of performance. Okay, so and it's not just unsupervised, unlabeled training examples. Those are examples that were painstakingly annotated by hand for the bounding box around every single pedestrian in those images. So this is unlikely to scale up to the more general problem of generic object recognition, in particular to the number of object categories that are available to us. So a back-of-the-envelope calculation by Irv Biederman several years ago, where he essentially counted the number of nouns on a couple of random pages of the English dictionary, and from that count extrapolated to the whole entire dictionary, he was able to estimate the total number of object categories to be roughly on the order of 30,000. So today those estimates fluctuates a little bit, but I think it's safe to say that anywhere between 10 and 50,000 categories is the total number of categories that are out there. And so if we think that we're going to need an army of mechanical Turk workers painstakingly annotating 100 million training examples for each of the 30,000 object categories, clearly we're going to run into trouble. So this is in stark contrast with the primate visual system. So what is interesting about the primate visual system is that it takes a very long time to mature. Development is very, very slow. It takes all the way through puberty for the visual system to be fully mature. That said, by age 6, a kid is typically able to name most object categories that are available out there. Okay? So the development is slow, it takes a long time, but once our visual system has developed, it's really very efficient at learning new object categories. So here's an example that I borrowed from a slide from Josh Tenenbaum, where they typically ask in the lab, they show one single example, a single positive example of a new object category. So the experimenter would tell you, well, here's a new type of bacteria from another planet. Here's one example. Look at the examples here on the right side and tell me which ones are likely to be other examples of the same species, for instance. So I could ask you, how much do you believe that this is an example of the same bacteria? What's your guess? Anyone? Oh, you guys are sleepy. So, okay, so yes, no, same, no. What about this one? No. What about this one? Yeah, probably. I mean, it's not like there is, obviously this is a very ill-posed problem. I've given you a single positive tuning examples, no negative examples. There's no right or wrong. What is amazing is that if you show this set of examples to, typical human observers, what's striking is the level of agreement across human observers in terms of stating what is a same example or belongs to the same object category or not. Okay, so it's striking to see how much people agree on that. And in particular, how much they generalizes from this single one training example, something that Chomsky and others have called the poverty of stimulus, which is how well we do so well from so little. So with that respect, I think biology has a lot to teach to machine learning and computer vision, which is specifically the point of this series of tutorials that I'm going to be giving today. So much of what we know about our ability to, yep. And by the way, I should say, please ask questions, especially if I use slang that I forgot to define. Yes?
I'll ask a question then.
Yes.
How old must a child be in order to know that if it's next to a cliff, it shouldn't take a step forward?
How long does it take?
Yeah.
Yeah, so you're,
The vision that's necessary for survival seems to happen long before the vision.
That's correct. So I wouldn't, don't quote me on the number, but I would suspect within about 24 months of age, an infant would be able to sense, a gap, say, on the floor. Or, so I agree. So and in fact, development is very complex. There's a whole hierarchy of developmental stages that unfold one after another, and it's quite true that visual object recognition and naming occur relatively late as compared to other visual processes and routines. That's a good point. All right, so most of what we know about our ability to recognize objects come from the so-called rapid visual presentation paradigms. So here's an example from relatively old work from the mid-70s by Molly Potter and colleagues. What I'm going to do is that I'm going to show you a series of frames, a series of objects, one after another. I'm going to be presenting these images relatively fast. Actually, let me flash them. These are flashed at a rate of 7 images per second. There is not enough time for you to scan images with eye movements at this speed. And in particular, given that the sequence is completely random, there is no way for you to exploit any kind of top-down expectations of any kind. At this speed, most of you would be able to detect objects, and in particular, you would be relatively good at memorizing most of those. So I could ask you, whether you saw an animal, yes or no, whether you saw, food items, etc. And human subjects are quite good at this task, even despite the speed of presentation of these images. So a slightly more modern twist to this task is what Simon Thorpe called in the mid-90s UltraRapid Categorization task. So I'm actually going to test you on this task. If you were in the lab, there would be discrete trials where an image is flashed, and then you would have to release a button and to indicate the presence of an animal. This is called a go, no-go task. Here I'm going to ask you to simply clap in your hands whenever you see an animal. Okay? So it's going to be a little bit faster than what you would see in the lab, but I think most of you should be able to do the task. So are you guys ready? All right, let's go. False alarm. Okay, so you guys are quite good. At this type of speed, it's not like you get every single details in those images. There's quite a lot of things. Okay, maybe I'm going to pause it because... Okay, great, thank you. You guys are very good subjects. So at this speed, it's not like you get every single details that are contained in those images. There's a lot of information that's not available to your visual system. But as you could see, most of you are actually quite good at predicting the presence or absence of an animal in those images. Moreover, I cannot do it here because of the refresh rate on the projector screen, but in the lab, as experimenters, we're able to present what is called an eraser mask right after the presentation of the stimulus. What this eraser mask does is that it really precisely constrains the amount of time available to your visual system for the kinds of operations that are needed for the task. If we present the mask just at the right time, it turns out that the stimulus would become invisible to people. And they would actually tell you, if you're an experimenter, that they don't see anything. But if you push them to answer and state whether they can detect the presence or guess the presence or absence of an animal in those images, they would be much better than chance, well above 90% of accuracy. Okay? There are hypotheses out there suggesting that what the mask is actually doing is interfering with the cortical feedback because of the close proximity between the stimulus and the mask, and that in turn, this interference with cortical feedback, which are needed for conscious perception, render the stimulus invisible. Okay, so what this task highlights is that it's possible to recognize objects in images without actually seeing the images, and perhaps through a single feed-forward sweep of activity through the ventral stream through the visual cortex. So what have we learned from this type of task? Obviously, those are not natural everyday vision where you are free to move your eyes, shift your attention around. They highly constrain the amount of time available to your visual system. And so what we're hoping is that these types of tasks are essentially tapping into what I would call the core object recognition system. It's essentially tapping in the kind of representation, very coarse-based representation that the visual system is building during the first 150 milliseconds of visual processing before cortical feedback become active. So what have we learned from these types of tasks? We've learned that this feed-forward sweep seems to be sufficient to enable rapid object detection. That's what I call the what problem, saying whether something is present or absent in those images. Interestingly, even on those images where people are actually correct at detecting the presence of animals, it turns out that there are a chance at localizing where the animal was. Okay? So feed-forward processing seems to be sufficient to answer the what problem, but not sufficient to answer the what is where problem. Yes?
Does it work also for non-animal pictures? Like if I would ask you to clap in the hands when you see a church, would that work?
So you're asking for other object categories. Yeah, so the rationale for the animal category is that ecologically and evolutionary there is probably something special about animals. We've evolved to survive predators and things like that. So that was the rationale, natural images and kind of ecologically valid class of stimuli. That said, over the years, several labs have replicated those experiments using a variety of stimuli. So it works just as fine with transport vehicles and many other object categories. So there is nothing special about animals per se. Okay, so as I was mentioning, the task is sufficient for the what problem, but not enough for the what is where problem. And moreover, as I'll show you in a couple of slides, after the break, it turns out that human observers are actually quite sensitive to the amount of clutter in those images when they perform the task. So typically people will be quite good when there is one dominant object in the scene, which is quite salient, when the amount of clutter is limited, but their performance drops quite rapidly when the amount of clutter increases. Okay. By the way, as a side note, for those of you who are in computer vision, I think there is an interesting parallel with the kind of tasks that we are routinely using in computer vision. So there are challenges for the WAT problem. Those are data sets like ImageNet, the Caltech 101, 256. And there are other challenges, so-called localization, I think they are called data challenges from, say, Pascal VOC, where the systems are required to actually draw a bounding box around the objects. If you look at the state of the art for these two tasks, I mean, until at least the last time I checked, those were quite different systems that were good at one and the other. So in particular, the infamous bag of words tend to be best at solving the WAT problem, while the part-based model with explicit spatial relationships across parts tend to be much better for the localization problem. So it's kind of interesting that it seems that there is this dichotomy between bottom-up and feed-forward processing, bottom-up and top-down processes in terms of solving these two tasks. All right, so back to computational models of visual processing. I think what these tasks have told us is that essentially they have asked us to revise kind of the classical models from the 80s. And so this is a very caricatural sketch of some of the old work by David Marr and later by Irv Biederman in the late 90s, the so-called recognition by components framework. And so without giving you too many details about these types of models, one thing that these models were essentially emphasizing was visual processes such as edge detection, grouping mechanisms, figure ground segmentation, detection of parts, and then relationships across those parts. The fact that people are unable to localize objects in those images, to me, suggests that this type of model is not able to account for the kinds of processing that's going on during this rapid categorization task. Instead, what I think those tasks are suggesting to us is that there is an alternative route that these processes may take. What I think they are suggesting, and I'll show you computational evidence later on for that, is that an initial bottom-up feed-forward recognition is possible from just a bottom-up activation of what I would call a loose collection of image features. Okay. After this first initial feed-forward sweep, it's then possible to start implementing more complex visual routines through interactions between bottom-up and top-down loops that would then allow us to solve additional parsing and scene interpretation processes, such as explicitly representing the spatial relationships across those features, figure ground segmentation, and grouping across cues. Okay. All right, so what I want to do for this three lecture, for the remaining of until the break, I want to give you an overview of a set of basic facts that have been accumulated over the past 40, 50 years of visual neuroscience. I want to give you a high-level view of what is currently known about the organization, the anatomy, and the physiology of one part of the visual system, which we believe plays a critical role in our ability to recognize objects, which is the ventral stream of the visual cortex. After the break, I'm going to essentially describe a computational feed-forward hierarchical visual architecture, which bears a lot of resemblance to a lot of the learning architecture that you've heard about in the past couple of days in the summer school. And I'll show you that a specific implementation of this overall class of models, hierarchical feed-forward models, is able to both qualitatively and quantitatively agree with the anatomy and the physiology of the ventral stream of the visual cortex. And I'll also show you that it's able to reproduce the level of performance of human observers during one of these rapid categorization tasks. And then tomorrow, during the third lecture, I'll describe a hierarchical Bayesian model, extension of this feed-forward model, that suggests how cortical top-down feedback signals could be interacting with bottom-up processes to allow for the implementation of more complex visual routines, such as attentional mechanisms and visual searches. Much of your brain is actually spent on vision. About rough estimate states that about 50 to 60% of your brain is devoted to vision, and that's a lot of neurons. Your visual cortex includes on the order of 10 billion neurons, about a little bit less for the monkey cortex, and I'm showing you here a monkey brain on this slide. So it's customary to assume somehow two independent streams of visual information in your visual cortex, a ventral WAT stream that seems to be mostly involved in the processing of shape information and invariant object recognition. This stream starts at the back of your head in the primary visual cortex and run laterally throughout the sides of your head here. And I'll tell you more about this stream in detail in the next couple of slides. This has to be contrasted with a dorsal visual stream, which runs, again, starts at the back of your head in the primary visual cortex and runs towards the top of your head. The dorsal visual stream, to a first approximation, seems to be mostly involved in the processing of motion information. And I'll show you more, I'll show you actually a computational account of that stream after the break. And also in the representation of spatial relationships across objects in visual scenes. And I'll integrate those findings tomorrow when we discuss attentional and top-down mechanisms. All right, so visual processing starts at the level of your eye, very briefly, because Bruno already mentioned some of those numbers yesterday. To a first approximation, you can think of your retina as a CCD matrix, 10 million, 10 megapixels for current CCD matrix, about 1 order of magnitude more photoreceptors. This is what your retina looks like. What you see here is an array of photoreceptors. Those are cones that you see. Those are sensitive to the amount of light that's imprinting them. So in a typical visual scenes, there is light bouncing off objects included in the visual scenes. Those various rays of lights get focused by the lens on your eye, and then they get to imprint individual photoreceptors. The photoreceptors respond in proportion to the amount of light, the number of photons that are imprinting them. Now, I'm not going to be doing much justice to the retina because there is quite a beautiful and intricate machinery going on at the level of the retina. In particular, your retina is already implementing several stages of visual processing. I'm not going to talk about it much today by lack of time, but instead I'm just going to take a shortcut and consider the output of your retina through the retinal ganglion cells. Bruno mentioned yesterday that there is about 10 million such fibers coming out of your eyes. Of course, there is a lot of debate that is ongoing in neuroscience about, specifically what are the meaningful signals that are contained, or the neural code that's being carried by neurons. But to a first approximation, many of us consider that the critical variable is the number of spikes emitted by maybe a single neuron or a small population of cells, maybe all tuned to the same visual feature over a short time window, something like 10 to 50 milliseconds. Okay? So just to give you a bird's eye view, here we see kind of this all or none events, but in practice, the spikes have some temporal extent on their own. They last about 10 milliseconds, just to give you a time scale at which these processes occur. All right, so as Bruno already emphasized yesterday, there's a lot going on already at the level of single neurons. Okay? There is already quite a complex biophysical machinery that's happening at the level of single dendrites. But to a first approximation, to a first order, it's possible to neglect some of these processes and assume a highly simplified first-order model of neural processes. So here we're going to be assuming that this ganglion cell receives input from a small population of 3 input units here. They are denoted X1, X2, and X3. We're going to be considering this level of activity perhaps reflecting the number of spikes emitted within a 10 millisecond time window from a population of afferent units. We're going to be denoting WI, the synaptic efficacy of synaptic weights of these afferent inputs, so far very similar to standard neural nets. And then we're going to be assuming a linear soma that's simply acting as an integrator and summing up those weighted inputs. And then we'll account for some of the nonlinearity of in particular, the conversion from membrane to potentials to spikes through a nonlinear function here. This can take a number of forms. I'll show you examples with half-wave rectification. You can have an hyperbolic tangent, many flavors. So how can we compute with these kinds of neurons? Sorry. How can we compute with these kinds of neurons? Well, for instance, a highly simplified version of that would be to simply consider weights as binary units, maybe either positive or negative. And if you implement something like that, you'll be able to approximate maybe something that looks like a center surround receptive field, where the center part of the receptive field will activate a excitatory set of subunits for this unit, and the surround here will be inhibiting the activity of the cell. That is decreasing in its response when it's being stimulated by light. Now, if you want to be slightly more accurate, there's been quite a bit of work in visual neuroscience suggesting that, at least starting in the visual cortex, one needs to take into account an additional stage, so-called divisive shunting inhibition, which is normalization by a pool of cells. So in the case of the primary visual cortex, this could be normalizing the response of one cell at a specific orientation by a pool of cells tuned to slightly different orientations, spatial frequencies, perhaps also over a different position in space, extending beyond the classical receptive field. All right. So to give you a flavor of the types of processing that are happening throughout these stages of processing, let's look at what's happening at the level of the primary visual cortex. So some of the early work was done by Hubel and Wiesel back in the late 50s. They got the Nobel Prize for this work in the 80s. And while recording in the primary visual cortex, they found several types of functional cells. So they found, and I should emphasize that the notations that I'm using here is slightly different from what I was using, where the pluses and minuses are now replaced with bars and crosses and triangles. So in addition to the center surround types of cells that people knew about all along that are found in the ganglion cells, both in the retina and the lateral geniculate nucleus, which acts as a relay between the eye and the primary visual cortex. In addition to those center surround receptive fields, they found evidence for cells tuned to different orientations. Okay? So here are examples of some of the cells that they found. You see a large variety. Some cells have a center excitatory, excitatory center flanked by inhibitory surrounds. Sometimes you could have the opposite. You can have things that act more like as bar detectors and some things more like wider bar detectors or edge detectors. So there's a large variety of receptive field organizations. So here's an example. Actually, I wanted to show you so this is an actual movie from Hubel and Wiesel doing one of these seminal recordings. What you see here is that they're using a fairly old apparatus, which is just a slide projector, where they're using the slides to present various kinds of bars and edges at various orientations. You should be able to hear a noise. The noise comes from the fact that the spikes that are recording through the amplifier is actually hooked up to a set of speakers, which allows them to hear the response of the cell as they are mapping out the receptive field. So what you see here that they are showing with those crosses is the excitatory part of the receptive field, which is the part of the receptive field that, if activated by a bright bar, may elicit a response from the cell. What they are mapping out here is the inhibitory subunit organization of this cell. If a bar of light is flashed in this subregion, the cell actually become inhibited. And one byproduct of this inhibition is that if you turn off the bar, as you might hear sometimes, the cell actually starts firing, which is a rebound from this inhibition. Okay? So here they are showing you the subunit organization of this cell on center of surround. They are now showing you that the cell is actually quite sharply tuned for orientation. When they present the preferred orientation, the cell responds very vividly, and much less so when the bar is rotated away from the preferred orientation. Okay? So let me skip through that. In addition to this population of simple cells, they also found a distinct population of neurons that they dubbed the complex cells. So the complex cells, much like the simple cells, are tuned to various kinds of orientations. So you see here an example of a cell that's tuned to a 135-degree orientation. However, what's striking about this cell is that unlike the simple cell that I showed you before, that has very distinct on and off subregions, it's not possible to find such subregions in the complex cells. Okay? So the complex cell responds in much the same way, whether the bar is presented at one location of the receptive field or another. Okay? By the way, in addition to this tolerance with respect to the exact location of the receptive field, of the preferred stimulus in the receptive field, they also found that these complex cells are actually invariant with respect to contrast reversal. So as much as I showed you that simple cells tend to be selective for either a white bar and a black background or the opposite, complex cells typically don't care about the polarity. So the same cell will respond to a white bar and a black background or the opposite. Okay? So here's an actual recording of a complex cell. As I mentioned, they are no longer able to isolate different subunit on and off regions. Instead, they are only trying to map out the overall receptive field. you see that the cell responds to the presentation of the bar at the preferred orientation anywhere within the receptive field. So that's one end of the receptive field. The other hand. All right. Okay, so what Hubel and Wiesel suggested is that perhaps a critical aspect of this organization has to do with hierarchical processing. And so their basic proposal, although qualitative, was to say that perhaps the orientation selectivity at the level of the simple cells could be obtained by pulling over the right kind of ganglion cells. Those are ganglion cells that are, for instance, aligned over an axis of preferred orientation. Okay? And then similarly, it's possible to build complex cells by pulling together afferent simple cells at different position and scale. Oh, sorry. Thanks for, can you hear? Okay, good. All right. Should I repeat this or? Okay, so the point that they made is that perhaps this type of organization could be implemented via selective pooling mechanisms. And so their suggestion was that orientation tuning at the level of simple cells could be obtained by pulling over afferent ganglion cells, the center surround cells, that are aligned along a preferred axis of orientations. Okay, and similarly, that it would be possible to build the complex cells by pulling together afferent simple cells all with the same preferred orientation, say a vertical bar, but slightly different position and scale. Okay, and I'll get back to that when I'll describe related computational models. All right, so beyond the primary visual cortex, there is plenty of evidence for these kinds of hierarchical organization. So this hierarchical organization is not just limited to the primary visual cortex. In particular, if you look at the organization of your visual cortex, Bruno mentioned yesterday that brain areas are organized, or the visual cortex is organized into 6 distinct layers. And we know that the way areas project onto one another depends on a very specific pattern of connectivity across those layers. So we know that the entry of a visual area is in layer 4, and we know that the output of a visual area is localized in more superficial layers. So bottom-up connections would project from superficial layers onto layer 4 of the next area, while the top-down signals would re-enter this area through deeper layers, such as layer 5 and 6. Okay? So if we look at the specific pattern of connectivity, and this is something that the anatomy has taught us across visual area, it has been shown that it's possible to recover an actual visual hierarchy. Okay? So this is an anatomical hierarchy. And this is shown here on the left, which is a diagram from Fenneman and Van Nessen. So what you see on this diagram is put at the bottom the lower visual areas. So this includes, starting at the level of the lateral geniculate nucleus, primary visual cortex. And then the visual hierarchy is run through a series of stages, and it culminates in actually non-purely visual areas, such as polysensory areas in the superior temporal sulcus, all the way through hippocampus, which is with cells that Mark Aurelio described yesterday as motivation for their Google brain, suggesting very sparse responses and very invariant responses to natural object categories. Okay, so the hierarchical organization in the visual cortex is obvious from the criterion of the anatomy. It's also quite obvious from the perspective of signal conduction and latency. So if you look at signal propagation through these areas, and so they are shown here on an actual monkey brain, as one goes in higher and higher visual areas, you see that there is an increased delay by about 10 to 20 milliseconds from one area to the next. Okay. Now, just a point of caution. Of course, there's a lot of noise. If you look at actual data, metadata analyses are typically pulled from study collected across many different animals, many different groups, many different types of stimuli. So if you look at the actual standard deviation around those mean values, they are quite huge. Okay. This also reflects the fact that we have quite a large diversity and variety of cortical cells. But if you look at the trends, and if you look at the median values, for instance, the hierarchical organization becomes quite clear, okay, with the latencies increasing at every stage of processing. And then most importantly, beyond this anatomical hierarchy and this hierarchy based on latencies, there's a clear functional hierarchy in the primate visual system. Okay? So I showed you that the primary visual system is selective for simple stimuli, perhaps the simplest kinds of stimuli, bars and edges at various orientations. Once you leave the primary visual cortex, people start finding tuning and selectivity for more and more complex stimuli. So here are examples of the kinds of stimuli that monkey electrophysiologists have been using throughout the years. You see that starting in area V2, one can find selectivity for something more complex than single orientations, like combinations of orientations. And then as you go up in higher and higher areas, one starts finding more and more complex preferred stimuli for the cells. So the representation culminates in the level of the inferior temporal cortex, IT, in particular the anterior part, where people have shown that neurons can be selective for object parts, or even in some cases, for instance, ecologically important classes of objects, such as faces and body parts, one can find cells which are very sharply tuned around some of those objects. So here are examples. I know it's a little bit hard to see, but those are examples of neurons tuned around specific views of faces. Okay? Now, in parallel to this gradual increase in the complexity of the preferred stimulus driving neurons, people have also documented a gradual increase in the invariance properties of neurons. Okay? So first of all, there is a gradual increase in the receptive field sizes. And I should point out that this slide is a little bit misleading because the brain here is mirrored by 180 degrees over this brain. So the back of your head, the primary visual cortex sits on the left here. And the higher visual areas, like the inferior temporal cortex, sits on the lower right corner. Okay. Nonetheless, you see that in early visual areas, like in the primary visual cortex, what you see here are receptive fields, their sizes, typical sizes. And receptive fields tend to be very small. Okay? So they run from typically a fraction of a degree up to maybe a degree or two of visual angles. So for those of you who don't know what a degree of visual angle is, this is the receptive field sizes unit that we use in visual neuroscience. A degree of visual angle corresponds to the width of my thumb at an arm length distance. So if you move around your thumb, you'll see that a 1 degree of visual angle really is a very small part of your field of view. Okay. So the processing at the level of these early stages is very, very local, limited to a very small portion of your field of view. And then as one climbs up this hierarchy, one starts finding larger and larger receptive fields. Okay? To the point where when one reaches IT, receptive fields can be quite large, spanning several degrees and crossing across hemispheres. Now, there are two words of caution that I would like to bring. The first one is that you'll find that the receptive field sizes that are being reported for IT vary drastically across groups. And that has to do with the way that they map receptive fields. In particular, it depends whether they are talking about the actual receptive field size, which for IT neurons can span several tenths of degrees of visual angle, or if they talk about the range of invariance of those neurons. That is, the range over which functionally the cell is able to treat similar stimuli over a range of position and scale as the same, as opposed to distractors. Okay. And those numbers tend to disagree quite a bit. So although receptive fields in IT can be several, say, tenths or 15 degrees of visual angle, the actual range of invariances is much lower, much smaller, on the order of 4 degrees of visual angle. So again, if you think about, if you tile the space with 4 thumbs, you'll see that 4 degrees is large, but it's not that large. In particular, that's not going to give you a complete invariance over the entire field of view. Okay. Just to give you an idea, there is typically about 120 photoreceptors per degrees of visual angle. So you should think that the range of invariance of these neurons is on the order of something like a little bit under 500 pixels across. Okay, so significant, but not complete. In order to build a complete invariance to transformations, then one needs necessarily to tile several of these receptive fields across space. Okay. The second point that I'd like to make, and this is related to a point that Bruno made yesterday, which is the fact that although I'm describing this hierarchy as a very clean, straightforward hierarchy, it turns out that, as always in biology, things are always a little bit less clean. And in particular, what I'm describing are coarse trends. But if you look at single areas, you'll be able to find relatively complex receptive fields already at the level of V1. Okay. And you'll be able to find relatively simple receptive fields, such as bars and edge detectors, all the way through IT. Okay. So overall, there's this trend for increase in the complexity and invariances, but I would describe that more as a loose hierarchy as opposed to a very strict hierarchy. All right. One critical component of cortical organization is the columnar organization. So yesterday, Bruno mentioned that within a cortical column of the primary visual cortex, there is on the order of 100,000 neurons. They implement a very redundant dictionary for representing images. They contain, each cortical column contains all possible orientations, multiple scales. One point that I'd like to make is that we shouldn't think of, or you shouldn't think of, cortical cells as doing one simple kind of detection of feature, but every cell participates in a multidimensional representation, which is illustrated here. So what I'm showing you here, and I unfortunately lost the source of this beautiful slide. If anyone knows, let me know what the original paper is. This is a optical imaging study. So optical imaging is a relatively crude way of measuring brain activity. Essentially, you open the scalp and put a camera just directly looking at the visual cortex. Okay, so this is a technique that works well for looking at the activations of cells on the surface of the cortex. And what's being used is a voltage-sensitive dye that will reflect different amount of light depending on the voltage of a patch of the visual cortex, which reflects the overall kind of population average activity. Okay. What you see here are shown in pseudo-color the actual selectivity of various patches of the visual cortex. So what you see here coded in color is the preferred orientations of various patches of the visual cortex. So you see a very clear organization in terms of preferred orientation, and this seems to be more or less stable across spatial frequency. But you can also look at this map now from the perspective of not edge detection and orientation tuning, but from the perspective of spatial frequency. And you can actually combine those maps into one map where you actually look at the preferred spatial frequency of cells or patches of the cortex, and you'll find a complementary kind of orthogonal feature map. So really, you should think of processing happening in the primary visual cortex and possibly all the way through the ventral stream of the visual cortex at these kinds of multi-dimensional analysis of the visual scenes along many different dimensions. And of course, I'm showing you examples with for spatial frequency, orientation tuning, but there is a lot of diversity in those cortical columns. So one can find cells tuned to various kinds of disparity across the two eyes, tuning for different hues, motion directions, et cetera, et cetera. And as I'll show you when we talk about computational models, one of the ongoing goal of the work in my lab is essentially to continue the extensions of these computational models to take into account this diversity and tuning across multiple dimensions of visual processing. Now, beyond the primary visual cortex, there is suggestive evidence for a similar cortical organization, but the evidence is rather scarce. So we have hardly any data regarding the organization of area V4 beyond orientation and simple tuning for U. Most of the work comes from a scientist at the Riken Institute in Japan, group of Tanaka using monkey electrophysiology and Tsunoda using optical imaging techniques. What you see here is a cartoon of the kind of organization that Tanaka has reported for the IT cortex. So what they did here is to show that as they penetrate in electrodes perpendicular to the surface of the cortex in IT, they tend to find neurons tuned to this similar kinds of visual features. Okay, so things like these kinds of visual features across orientations, various kinds of family of simple shapes. Again, I'm not sure this is necessarily reflecting the reality and should be taken more as a cartoon. If conversely, if someone penetrates more tangentially the visual cortex, then one finds a very discrete kind of transition across cortical columns where suddenly the selectivity of neurons changes quite abruptly. This is a similar example, again, from optical imaging, where you see that essentially what one should think of the representation of a single stimulus like this one as activating various kinds of patches from this area IT, whereby every single patch would code for different visual features from this stimuli. Okay. So I'll get back to that when we discuss the computational models. All right. The point I'd like to make is, there is a series of processing going on in the ventral stream. By the time one reaches the higher level areas, such as the anterior inferotemporal cortex, AIT, I would like to claim that the problem of invariant object recognition is essentially solved. So this is a little bit of strong voice, but let me show you a study that was conducted a couple of years ago at MIT in the lab of Jim DiCarlo. So they recorded from a small population of about 200 neurons in the inferotemporal cortex. They considered a small set of 77 stimuli that they organized into more or less arbitrary classes of objects. So they tried to have something that was meaningful for the animal, things like the faces of the experimenters or the handlers for those animals, the kinds of toys that they had available in their cages, the kinds of food they eat, body parts, etc., etc. So 77 stimuli falling each into 8 distinct object categories. And the experiment they did was to essentially ask the following question. If we consider a small population of about 200 IT cells, if we consider their firing rate over a short time window, say about 20 milliseconds right after the neuron starts firing in this area, and if we pass this rate code to a linear classifier, the simplest kind of classifier one can assume, a linear SVM, how well could we predict the object category of that was presented to the animal? One point that I should emphasize is that in those experiments, there was no task. So the monkey was just passively fixating on the screen. So here's what they did. Essentially, they considered these 8 object categories and trained a linear classifier on a subset of the stimuli and tested the linear decoder on disjoint sets of objects and showed that indeed it's possible to very reliably decode the category information related to the presented stimuli at a relatively high level, about 80% of accuracy, when chance level would be somewhere around 12-13% of accuracy. Okay. Now, more interestingly, what they showed is that it's possible for this linear decoder to be trained when objects are presented at one location and scale. Okay. And from training at that one location and scale, the classifier was tested at different position and scale. Okay. And what you see here is that although there is a small decrease in the decoding performance, the classifier is still able to predict the category of the objects irrespective to these changes in position and scale. Now, the reason why they really tested for the generalization of the classifier is that, of course, if they were to train a classifier, even a linear classifier, on a stimuli at multiple position and scale, it could have been that the linear classifier would have learned the change of appearance of these objects, right? The fact that the classifier never saw those examples as training suggests that the invariances that are being required for performing the job are built in at the level of IT already. Okay, this is not something that's being learned by the classifier, but instead the representation as this built-in invariance. Okay. So I think this is suggestive evidence that you can do already quite well in terms of decoding invariant object category from a very small population of IT neurons. So IT contains on the order of 10 million neurons. In comparison, this is a very, very small subset of about 200 neurons. So the claim would be that just by scaling up the number of neurons, we would be able to reach something close to human level by considering a population that's large enough. Again, untested. Okay. But as I emphasized, this is actually still there's no task. The monkey is passively fixating, and it would be nice to be able to relate this category selective neural activity to an actual behavioral response, right? So it's possible that the information is there, but it wouldn't be used by the visual system in any way. So we have actually started a collaboration with a monkey electrophysiology group in France. This is work done by, in the lab of Denis Fils. A grad student who did the recording is Maxime Cauchois. And what they did is train a monkey to do one of these rapid non-animal categorization tasks. So this is actually a case, you probably can't see anything because this is an actual pretty tough version of that where we present an eraser mask right after the stimulus. What you see here is that the monkey is responding by releasing a button press here and touching the screen. Okay. What you see here is the neural activity that we record while the monkey is performing this task. Okay. So you see the monkey here. We have electrodes scattered throughout intermediate to high visual areas across V4 and PIT. And what you see here is those are surface electrodes. So what we record are the so-called local field potentials. We don't record spikes. Instead, we record the sum activities over populations of about 100 to 1,000 neurons. Okay. This is more akin to EEG. Some of you have heard about it, but sitting right on the surface of the cortex. So to my great surprise, the monkeys were actually quite good at doing the task. So what you see here is that on a familiar set of images, they were very close to seeing performance, well above 95% accuracy. So I was actually pretty skeptic about that, and because they use a relatively small set of about 300 stimuli for the monkey, I suspected that maybe the monkey was just memorizing individual examples. So what we did is give them, ship over the stimuli that we were actually using in the lab on human participants. So this is a completely disjoint set of images. And they tested the monkeys on the very first presentation of this new stimuli. So those are completely new stimuli. Those are stimuli that the monkey had never seen before. Okay? And I should emphasize that even from the perspective of computer vision, the task is quite difficult because of the large variety of animals that we consider. There could be anything from insects to mammals, all the way to fish, et cetera, et cetera. So very, very challenging task. What you see here is the level of performance of those two animals. So each bar color corresponds to one of the two monkeys we considered. So there is a small drop in performance, but they are still able to generalize to new previously unseen stimuli. Just to give you a baseline performance, this red bar corresponds to different kinds of monkeys. Those are MIT undergrads that we tested while I was a PhD student there. It turns out that the monkey are actually slightly better than human observers on the task. Not only are they more accurate, but they're also much faster. And this is mostly due to the fact that their brain is much smaller. So just because of conduction delays, smaller brains tend to transmit information much faster. Their brain is about 50% smaller, and they are about 50% faster on the task. Yes? Let me just skip to the, just not to hear the noise of the.
How does the monkey coding activity has concepts of all the animals?
Sorry?
The monkeys you use, were they captured in the wild or something? Because
No. So those are.
they have a concept of all these animals.
Yeah, so those are monkeys that were trained. So those are lab animals. Those are not zoo animals, but they were trained. So,
So were the pictures of the same animals in the test set? And they were...
Right. So that's what that's the point that I was trying to make. I was as skeptical as you are, and if you look at this is the performance on a familiar set of images, 300 images, okay, that the monkey saw over and over again. The performance here is on a completely different set of images. This is a set of images we're using at MIT when we're doing human psychophysics. So they never saw those images before. I mean, there might be some overlap in terms of the kinds of animals, but there is probably a lot of images that, animals that were, presented in the test set and not even present in the training set. So, to me, this is evidence that they were able to capture, a high-level percept or concept of animal category. Okay, so we conducted a similar decoding experiment. So we considered the neural responses or the field potential activity through this this dozen of electrodes. And I'm realizing that I'm running out of time. So what you see here is the performance of a linear decoder in terms of predicting whether an animal is present in every single image presentation or not. What you see here is chance level computed using a bootstrapping procedure. The widths of the curves here already correspond to the confidence interval that we derived using cross-validation techniques. So there are a couple of points to be made here. The first one is that you can see that we can decode animal category information very, very early. In fact, even earlier than what people have been able to decode during this passive viewing conditions that I described earlier on. Okay, so starting at around 70 milliseconds, we can already reliably say whether an animal is present or not. Okay. This could be due to the fact that we are recording from, our recordings include earlier areas which potentially contain relevant information for the task and for which the latencies are shorter. Now, more interestingly, I think this experiment is actually the first suggestive evidence of an actual involvement of this feed-forward sweep of activity, this very early categorical activity onto behavior. And so the way we establish this link is as follows. We subdivided trials according to the reaction time of the animal. Okay, so this is not considering any kind of neural activity. We simply look at quartiles for which, depending on the reaction time of the animal. So on the far left here are the darkest quartiles correspond to the fastest trials, while the rightmost quartile here correspond to the slowest trials. And you see that there is a nice kind of distribution of reaction time. What we did is to conduct the decoding separately on these four kinds of trials. And what you see here is that pretty much on the fastest trials, when we know that the monkey will respond faster, we can decode category information much faster than on the trials that for which the monkey is slower. Okay, so in other words, there's a direct mapping between the speed at which we can decode visual information in this ventral stream and reaction time, which suggests that perhaps there is a direct link between those two. Of course, what I'm showing you is correlation and not causation, but I think given the evidence we have so far, it is quite suggestive. Okay. All right, so let me see. Let me very briefly spend 5 minutes. I just because a lot of you have, or you spend the past couple of days hearing about learning rules and machine learning, etc. I at least wanted to mention something about learning and plasticity in the visual cortex. So to disappoint you, we know very little about the specific learning rules that could be implemented in the visual cortex beyond the obvious, which are related to things like Hebbian learning rules and spike time dependent plasticity, which are much more at the mechanistic cellular level rather than at a high level, kind of computational level learning rule. However, at a kind of course level, I think it's fair to say that it seems that learning and plasticity follows nicely from this hierarchy. And in particular, it follows nicely from criteria of stability. So if you look at evidence for learning and plasticity in the primary visual cortex, it's typically very, very difficult to elicit for experimentalists, and there is debate about the extent to which there is actual learning and plasticity. If you look at intermediate stages, it's a little bit easier and there is a little bit more evidence. By the time you reach higher level visual areas such as IT or prefrontal areas, there's actually quite good evidence for very fast learning and plasticity in those areas. Okay? So for instance, there's a paper that was published a couple of years ago in the group of Jim DiCarlo. Although the problem was about learning, the emphasis of the paper was about learning of invariances. I take this paper from a slightly different perspective, which is the fact that what they've shown essentially is that just by messing around with the visual experience of the monkey for 10 minutes, so what they did is that they essentially asked the monkey to fixate on objects, and then they would flash the same object in the periphery, and the monkey was trained to saccade to the other location. Both stimuli were falling within the receptive field of individual neurons in IT. Okay, and so if originally, say, the monkey was very strongly or liked the stimulus very much, it was a fairly good stimulus for the neuron. The neuron would fire, very strongly to the two locations. Okay, what they've done is played a mean trick to the monkey, which is while the monkey would be moving its eyes, they would swap on half of the trials the identity of the target. Okay. So there are mechanisms in the visual system to actually render you blind to these kinds of eye movements. And this is to suppress the motion cues that are associated with your eye movements. So I can play this trick on you and you wouldn't even notice that I did anything. Okay? Except that when you land on the new target, the identity of the stimulus has changed. What they've shown is that just playing this trick on the monkey for 10 minutes, they were able to essentially change the selectivity of the neuron. Whereby now if you swap between a good and a bad stimulus, the neuron would tend to associate the bad stimulus as a good stimulus. Okay. And to me, that means that essentially our IT cortex is changing all the time as we experience the visual world, which is almost a puzzle. So of course, this makes sense from the perspective of learning in hierarchies, right? Because if we were to have very fast plasticity in lower stages, we would have to relearn everything all the time, as opposed to if we have a long developmental stages where things are done Hinton style in a greedy manner, one layer at a time, where once you've learned a good representation here, you stop changing it and start updating in higher and higher stages. What that would allow you to do, presumably, is to exploit unsupervised learning mechanisms in lower to intermediate visual areas and enable very fast learning and plasticity, supervised plasticity in higher stages. There is actually evidence for supervised category learning in fairly high, non-purely visual areas, such as prefrontal areas, the prefrontal cortex. There is work by Earl Miller, using this kind of morph stimuli, and the use of morph stimuli allows them to control relatively well for the visual similarities across visual stimuli. What you see here is the response of a so-called category-tuned neurons after the animal was trained to distinguish between cats and dogs. So the important fact about this neuron is that you see that although from one line to the next here all the way down, the visual similarity of stimuli is moving by constant steps from something like 100% dog and 0% cats all the way down to 0% dog and 100% cat. So although the visual similarity or dissimilarity in terms of the appearance of this individual stimuli is constant, you see that the neuron has essentially distorted the space and essentially now treats, after training, all of this dog stimuli pretty much as the same versus all these cat stimuli as the same, and essentially stretches the space around the boundary when we go from a 60% dog to a 40% dog, which would be equivalent to a 60% cat. Okay. All right, so let me skip that in the interest of time. So just to summarize, what I've tried to do during this first hour is to give you an overview of what is currently a set of well-accepted facts about the anatomy, the organization, and the electrophysiology of the visual cortex. I've tried to highlight the fact that one striking feature of the organization of the visual cortex is its hierarchical organization, and that should sound familiar to many of you here in this summer school. The second point that I try to make is that we've learned a lot because of this rapid categorization task. So although they are highly non-natural, I believe that they tap onto what I would call the core object recognition system, the kind of base coarse representation that the visual system builds before more complex visual routines can be deployed. And I've made the argument that this can be well approximated by a single feed-forward sweep of activity throughout your visual cortex. Okay. All right, so let's take a break, and then when you guys come back, I'll show you an overview of, or at least I'll try to highlight how a specific implementation of a feed-forward hierarchical model of visual processing is able to explain a host of visual phenomena throughout the visual cortex. Thank you.